Hello dear students. Now today we are going to carry the demonstration for spot number E, that is to study the separation of plant pigment by paper chromatography. For that, first of all we will see what are the requirements. The requirements for the same are: one is a chromatography jar. You can see here the chromatography jar. This is a chromatography jar with its lid. This is a lid. And in this is a chromatography paper. Now more closer, this is a chromatography paper which is loaded with a chlorophyll pigment. So two things: one is a chromatography jar, this one. Second is a loaded chromatography paper. And the third one which we need is a solvent. Here I have prepared the solvent. These are the major requirements for this experiment of separation of chlorophyll pigment. Now what we have to do, we have to prepare a solvent. Solvent is prepared by using petroleum ether and acetone in 9:1 proportion. That is, if you are taking petroleum ether 90 ml, you have to take 10 ml of acetone, and that will be the solvent which you are going to use for separation of photosynthetic pigments. Okay. Next one is how to prepare the chlorophyll extract and how to load it. Now for that, what you have to do, you have to take 4-5 fresh spinach leaves, clean, wash it, dry it, and then make a paste using mortar and pestle in acetone. Acetone acts as an organic solvent. You have to make for the separation of photosynthetic pigment, you have to make the paste of the chlorophyllous material. We are taking here the spinach leaves using the acetone. Now why to use acetone? It is the organic solvent. Along with that, while making the paste of spinach leaf, you are going to add MgCl2, magnesium chloride. Just a pinch of magnesium chloride, it acts as a buffer. And you will get the filtrate. Whatever the content you will get, you have to filter it using the filter paper. And the filtrate is then either condensed or sometimes without, if it is thick enough or dense enough, you can load it on a chromatography paper using the capillary. Now here, in this chromatography paper, here, this is a complete, the chromatography paper is cut in a strip like this. In a strip it is cut. You can see here the strip of a chromatography paper. In this way, chromatography paper is cut into a strip. At the tip, a wick, it is cut into a wick. And in the middle of a wick, a pencil spot is given on which the condensed extract of chlorophyll A is loaded with the help of capillary. Now, how to load the chlorophyll pigment? You have to put a drop of condensed chlorophyll using the capillary, then let it dry either in the natural sunlight, or you can use the heater. You can use the dryer at lab. नॅचरल आर्टिफिशियल कंडिशन्समध्ये तुम्ही ड्रायर वापरून सुद्धा त्याला ड्राय करू शकता. प्रत्येक वेळी स्पॉट ड्राय झाल्यानंतर त्याला नवीन स्पॉट त्या ठिकाणी लोड करायचा. सो आफ्टर एव्हरी लोड द स्पॉट इज ड्राईड विथ द हेल्प ऑफ ड्रायर आयदर और इन अ नॅचरल heat or sunlight, natural sunlight. In this way, the chlorophyll pigment is loaded 10 times and ultimately you get a loaded paper. This is your loaded chlorophyll pigment, loaded paper, chromatography paper ready for the experiment. Okay, now let us start with the experiment. What we have to do is, we have to add the solvent in the, we'll keep the chromatography paper aside for a time and we will take the chromatography jar. Now we'll add the solvent in the chromatography jar that we have prepared. What it is? It is petroleum ether and acetone in 9:1%. And we have to add the chromatography paper in such a way that the loaded spot should not get immersed in the solvent and only the wick get immersed like this. You can see, and as you dip it, immediately the solvent start running in the upward direction. See that the your loaded chlorophyll pigment should not get immersed into the solvent. Then put the lid and allow. Now see, allow the solvent to run across the. Now you can see at the base the solvent, and you can even see the solvent is running in upward direction. When the solvent runs in upward direction, it carries the different chlorophyll pigment which are concentrated here in the loaded spot. It carries upward, and when the solvent runs along the chromatography paper, the pigments are separated. The pigments are separated on the basis of the molecular weight. And when this solvent, now you can see gradually the solvent is moving upwards. Now let it move up to the another end. Then take out the paper. Allow it to dry either in sunlight or even once again you can use the dryer, and you will observe that 4 pigments get separated as chlorophyll B just above the loaded spot. You get the chlorophyll B separated in the form of yellowish green patch. Just above chlorophyll B you get separated chlorophyll A in bluish green color patch or spot. Then above chlorophyll A you will get the yellow patch of carotene, and at the top you will get xanthophyll separated in the which appears in orange red color. In this way 4 different pigments get separated in the order chlorophyll B, chlorophyll A, carotene, and at the top xanthophyll. This sequence you have to keep into mind, and in the conclusion or in the result or rather in the comment you have to write this, because this is a demonstration experiment. You need not have to perform the experiment. Okay, so you are not performing the whatever the content, whatever the solvent we are preparing is prepared, that is prepared by your teacher. Then the loaded paper will also be prepared by your teacher, and you will get just a demonstration. Then what you have to do, you just have to identify the experiment from the experimental setup. The experimental setup will appear like this. What you can see in the video as a chromatography jar, chromatography paper inserted and with a loaded spot at the base, and the solvent added in the chromatography jar, and solvent is running in upward direction. In this way you can come across the experimental setup, which will give you an idea that this is the experiment for the separation of plant pigments or photosynthetic pigments by paper chromatography. So, and that will be the identification. In this spot, you have to write the correct identification, you have to write the correct comment. Your correct identification is, this experiment is to demonstrate. डेमॉन्स्ट्रेशन एक्सपेरिमेंट आहे म्हणून आपण म्हणतो की हा एक्सपेरिमेंट काय डेमॉन्स्ट्रेट करतो, इट डेमॉन्स्ट्रेट separation of photosynthetic pigments by paper chromatography. When you will write this one, this line, you will get the weightage for correct identification, that is half mark. And when you write the conclusion or inference, that will be the comment, that how many pigments are separated in which color and which sequence. That is the comment. So in the comment, what you will write your observation, that after separation, when the pigments get separated, chlorophyll B is found separated just above the loaded chlorophyll pigment in yellow green color. Above chlorophyll B, chlorophyll A gets separated in blue green color. Above chlorophyll A, carotene gets separated in yellow color, and xanthophyll at the top is separated in the orange red color. So that will be your comment, and this is simply a demonstration experiment, and you nothing you have to do ॲट युअर ओन अकॉर्ड. तुम्हाला स्वतःला स्वतःहून कुठलाही प्रयोग किंवा त्याच्यामध्ये कुठलेही ॲडिशन्स असं काही करायचं नाही. हा डेमॉन्स्ट्रेशन एक्सपेरिमेंट आहे. ऑब्झर्व करायचा, ऑब्झर्व करून एक्सपेरिमेंटल सेटअपवरनं आयडेंटिफाय करायचा. आयडेंटिफाय केल्यानंतर त्या एक्सपेरिमेंटचं नाव लिहायचं, दॅट इज युअर करेक्ट आयडेंटिफिकेशन, आणि नंतर मग त्याचं ऑब्झर्वेशन किंवा कन्क्लुजन जे आहे ते लिहिणार तुम्ही. दॅट विल बी युअर कॉमेंट अँड यू विल गेट 1 मार्क. दिस इज फॉर spot number E, one of the demonstration experiment for spot number E. Another one more experiment is also there, that is a 4-leaf experiment. Rather more correctly we can say separate study of rate of transpiration by 4-leaf experiment. The demonstration of the same we'll see in the next video, and for this demonstration we'll stop over here. Thank you very much. Shall we stop?